excited to tell you a little bit about a research project that I started several months ago. This could be subtitled, What I Spent My Summer Vacation Doing So Far. This is a collaborative project with some folks in Brisbane, Australia, at Sugar Research Australia, and I was asked to come work with them on a very large-scale data analysis project involving RNA-seq data from sugarcane. The main deal here is that they have detected a new emerging syndrome called Yellow Canopy Syndrome, or YCS, in sugarcane, and they don't know what causes it. And so today, I will be helping you think a little bit about the motivation for this particular project. YCS has only been around for a few years, first detected in 2012. It's mostly located in Queensland, in the northeast coast of Australia. It's characterized by a fairly unusual leaf yellowing. It doesn't reflect any known patterns in sugarcane syndromes or diseases. 2 years following the initial appearance of it, they detected particular patterns, or more appropriately, a lack thereof, in which they found most cane growing regions in Australia affected, but the symptoms appear and then disappear in waves. The ultimate result of when the syndrome appears is that crop growth and subsequently yield may be reduced by anywhere from 30 to 70%. Given that sugarcane is a 2-year crop, this means that the farmers are very interested in making sure that some sort of resolution is determined. Before you can resolve the syndrome, though, you need to figure out what causes it. And so there is a number of different approaches SRA has been taking to uncover this initial causal agent, including general physiological assays of sugar and starch, also metabolomics, metagenomics, and then transcriptomics, which I'll be telling you a little bit about today. Some of the hypotheses we have for what may be causing YCS may actually be abiotic factors. And so, for example, we have looked at things like nutrition, various types of environmental stress. In that part of Australia, there's been pretty serious drought and heat stress over the last few years. The presence or absence of herbicides, and then various types of root growth deformities have all been assessed. The biotic factors that we've been investigating include insects, which may cause problems in the plant itself or may be acting as vectors of disease, fungus, bacteria, including a number of known phytoplasma that affect sugarcane, and my particularly favorite hypothesis, the presence of some sort of virus, including a retrovirus or a pararetrovirus, that may have emerged from the genome or been brought in from somewhere else. All of these have been assessed at the various standard assays used to detect pathogens or abiotic stress in the group, and all of them have been found negative, according to most of the standard approaches. We've undertaken a transcriptome study because each of those possible causes has a set of genes that we've determined or expected to be differentially expressed in our data when comparing control and syndrome-affected plants. The data that I was brought in to help with are quite hefty, if I do say so myself. What this diagram shows you is a overview of the different populations. FV stands for field visit. Each of the numbers stands for a different year in which these tissue were collected. These have also been collected across a number of different regions within Queensland. We have collected both control and YCS samples for most of these populations that have been collected with the utmost attempt to control for all possible variation. So samples collected from the same field, both affected and non-affected leaf tissue. We have gone through and sequenced 4 taxa per lane, paired in sequencing 100 base pair reads. And by applying a stringent filtering for data quality, we managed to reduce the data set quite a bit. I actually don't know how big the unzipped or uncompressed raw data are, but after trimming for filtering, it's still almost a terabyte worth of data, which is a bit of a bear to analyze. We have normalized these reads across each field visit, or each what I'll call a population. And then the final data set that I've been analyzing for assembly is about 142 gigs worth of data. The methods I've been using for assembly include first normalizing in silico all the reads across across the entire data set, that 142 gigs worth. And then using Velvet with Oasis merging, Soap de Novo 2, Soap de Novo Trans, and Trinity. Each of these have different levels of k-mers that we've been assessing, described there. The approach here is to make sure that we're capturing transcripts at different lengths. And the results, not too good right now, not gonna lie. We have way too many contigs, more than we would expect. We're expecting somewhere between 80 upwards of maybe 100,000 transcripts or genes. I've been getting way more than that. I've been applying different merging algorithms using evidential gene to reconcile the SOAPs, which are not automatically merged within the programs. I've also been playing around with CD hit to try and collapse those. Today I'm going to be talking about the results I obtained from Trinity that has been subsequently merged within itself through evidential gene, which of course is not recommended by that actual software developer. But I do end up getting only 225,000 contigs with a pretty abysmal N50. Regardless, I want to have something to look at, something to help me optimize this data set, so I persevered. The reason I persevered is that sugarcane actually ends up being a really difficult genome to work on. Cultivars and sugarcane have variable genome sizes. Generally, they tend to be about 10 gig bases. They also tend to be polyploid and often aneuploid. Sugarcane, as we know it as a crop, is actually a hybrid between Saccharum officinarum, which is an octoploid with 80 chromosomes, and Saccharum spontaneum, which again has quite variable chromosome numbers and ploidy. The complicating factors of this particular data set include possible contamination from pathogens that may be present. I put contamination in quotation marks because, again, we want to find the source, so I actually have not filtered out any of those bacteria or viral sequences, and also estimating 8 to 10 alleles per gene that may be possible. And so what I'm considering doing, and I'm still working on, is going back and filtering out that contamination, and also using a low stringency clustering of contigs, which has been done in a couple of other sugarcane transcriptome studies, to try to get a more manageable set of transcripts. My quantification and differential expression approaches right now, I gotta give a big old plug out there for the Pasture Lab, who have been developing these amazing methods. They have Callisto, which is named after the Greek myth, and then Sleuth, which on their website it cracks. Has anybody used these before? If you go look on their website, they're like, in the FAQ it says, why is it named Sleuth? Is there a reason for it? And the answer is yes. I'm like, great, okay, you're already my friends and I don't even know you. The way Callisto works ends up being really nice for my particular purposes. This is a read quantification program that does not use alignment of reads. It uses what's referred to as a pseudo-alignment quantification that ultimately ends up being fast and, compared to other transcript abundance methods, fairly accurate as well. Callisto is paired with Sleuth, which is an R package designed to integrate with the R package Shiny to help you explore visualization and do basic differential expression analyses associated with the Callisto abundance estimations. The visualizations I'm showing you today are all out of the box directly from Sleuth, so they're not the best visualizations, but give me some talking points for this. My differential expression approaches here include both a volcano plot, which indicate the significant genes highlighted in red with a max FDR 0.1, and then a QQ or quantile plot here that help you look at sort of the distribution of those particular transcripts. Remember, I have almost 225,000 transcripts. So we have some things that are differentially expressed. These populations are not across the entire data set. This is a single population comparing 10 control and 10 YCS samples. Some of those most highly expressed genes or show the most variation between our data set do include some bacteria. Ralstonia and Acidovorax, which are both known as crop pathogens, in fact, have been found in our data set, and they are differentially expressed. However, these are also known as soil-associated bacteria, right? So again, something you might find growing in a sugarcane field. As far as the sequences we find that may be actually from our plant, we do get some significantly expressed genes from both sorghum and saccharum. However, they all annotate as hypothetical proteins. This may be a common issue to a lot of you, but I'm still beating my head against it. Moreover, those bacteria that I just showed you are differentially expressed, but those genes actually have lower expression in our affected YCS samples. So here's a heat map showing you on the y-axis A through F, the most highly expressed or highly differentially expressed genes from bacteria, with YCS here and control here. Again, red means a lower expression. What's nice about this is that I can see I actually have 2 samples, 1 and 6, that stand out as a little bit of outliers from the rest of the data set, which may help me go back and filter, again, because the syndrome is so difficult to diagnose in the field, may help me filter the data a bit more to detect a more effective signal. Where I'm planning on going from here, and at this point I have to cry for help and ask for any recommendations anyone might have for helping me sort out this problem a little bit better. Now that I know what some of the bacterial sequences in my sample might be, I'm thinking of going back and screening the entire data set prior to assembly for contamination, using those bacterial genomes that I found to be associated with these samples. I'm also planning on doing a bit more optimization of assemblies using longer kamers for soap in particular, which may help me reconcile some of the very short contigs I found in the data set, or using different clustering algorithms. Other folks in sugarcane in some cases use an 80% threshold in CB hit, which seems really low for me, but at the same time, if you're dealing with 8 to 10 alleles, it might be warranted. I am really excited about going back through and using the full power of this experimental design to figure out what the variation in our expression data are telling us. And then finally, I'll be hitting the transcript annotation pretty hard over the next few months. In particular, using domain searching for both transposons, if it is associated with some sort of pararetrovirus, what's happening with those repetitive elements, and figuring out what some of the protein genes are doing. Finally, I'd like to thank all of my collaborators. I couldn't have done this analysis without the Texas Advanced Computing Center here in Austin, where I do all of my analysis, as well as data card transfer carpentry. I do teaching for these organizations because I initially learned to do all these analyses from them. My collaborator is called Kate too. I call her the other Kate because I'm also Kate. So here are the 2 Kates. I just got back from Australia a few weeks ago from working with them. You are free to tweet at me if you have any recommendations, or tell me right now what it is you think that I should do. Thanks. Yes?
So you're getting a lot of uncharacterized things. How are you doing your annotations?
We've been using a basic BLAST. First off, because we have so many contigs right now. We've been using, my collaborator likes BLAST to go. Yeah.
So I've been doing annotation of weird things by basically sorting into existing groups of orthologous groups and pulling those annotations
All right.
for things where you don't have a direct link to BLAST.
Fantastic. Okay, so I'll hit up the ortholog annotation a bit more.
Good ortho MCL and eggnog.
Okay, awesome. Like I said, I want to get the number of contigs down a little bit first because it just ends up being so difficult to run the analysis in the first place. Is the orthology analysis pretty fast?
Yeah, a few days.
A few days? All right, I can handle that. Anything else? Yeah.
I'm just curious about the differential abundance of the bacterial transcripts. I know you said
Yeah.
the bacteria were typically pathogens and that they were lower
Yep.
expression in your samples. But
Yep.
I don't know how closely those pathogens are related to things that aren't pathogens, and if it's possible
Right.
that actually you're seeing a loss of beneficial bacteria that could be a secondary effect.
Sure. And I think part of the problem is that I should probably just go back and pull out all the data. And maybe we can talk about this later too, Erin. Thank you. The next